Hey guys, welcome to Advanced Biology, Chemistry of Life, Proteins and Nucleic Acids. All right, so we're going to talk about proteins and nucleic acids today. And as I say, proteins are polymers made of the monomers called amino acids, and amino acids do make up proteins. So all the proteins that we talk about in just about every single living organism are made up of of 20 different amino acids. Okay, so there are literally thousands of amino acids out there in the world, but all living organisms only use 20 of these thousands of different amino acids. We use the same 20. It's one of the biggest pieces we have for the unity of all life, meaning there's a common ancestor to all living organisms on this planet. Okay, and proteins have a lot of different functions. I like to call them the workhorses of our bodies, and they make up a large portion of our dry weight. What do I mean by dry weight? If we took all the water out of our bodies, of course, we'd be dead by then. But if we dried ourselves out and we weighed up what was left, the biomass that was left, about 50% of that would be made up of proteins. And there'd be other things like some nucleic acids, some carbohydrates, and some minerals like calcium from your skeleton and things of that nature. But proteins have lots of different functions, and we like to group them by function. Structural is one of those, and I have this picture of this actress up here, and we can see her lips are all puffed up because she had a collagen injection. Collagen is an elastic-like protein that gives our skin its elastic abilities. Other proteins are contractile, and you can see we have a sprinter down here. And those are our muscles, basically. They're long proteins that slide past one another. Other proteins act as storage molecules, which basically we see in lots of seeds and things of that nature that store various types of enzymes and things that they need to grow. Some are hormones, okay, things like insulin, and they act as cell signals in our body. Of course, one of the biggest and most important roles that proteins play in our body are enzymes, and enzymes are biological catalysts. What's a catalyst? A catalyst speeds up a chemical reaction. Our bodies are relatively cold, 37 degrees centigrade, 98.6 degrees Fahrenheit. Okay, that is pretty cold for a chemical reaction to occur. It would occur very slow. In the chemistry lab, we can heat it up. We can add heat to it, bring it up to 200 degrees, bring it up to close to 100 degrees centigrade, 200 to 12 degrees Fahrenheit, and those reactions will occur very quick. As a living organism, we don't deal well with those high temperatures, so enzymes play the role of heat. They act as a catalyst. They make the reaction go faster, and they make it go really fast. And another big important role are cell membrane transport proteins. These are proteins that are going to allow the movement of different types of molecules in and out of the cell. These molecules are not able to move through the cell membrane, but they can go through a transport protein. Many diseases and disorders are a result of misfolded or in non-working cell transport proteins. All right, so all proteins are made up of amino acids, like I said earlier, and all amino acids have the same basic structure. They have this nitrogen, carbon backbone. One end of the amino acid has what we call an amino group. Okay, and the amino group is a nitrogen, hydrogen, and that can act as a base, and we'll get into that a little bit later. The other end has a carboxyl group, and a lot of times we like to write this as COOH, and that's the carboxyl group. What it is a carbon bound to an oxygen and a hydrogen. That's called a hydroxyl group, and another oxygen bound by a double bond. So all proteins have this basic structure. An amino group, a central carbon, the carboxyl group, that is the basic structure that all amino acids have. What makes the 20 amino acids different is this side chain called the variable R group. Okay, it's called the variable R group. This side chain is what gives each amino acid its specific properties. Is it polar? Is it nonpolar? Does it act like an acid? Does it act like a base? And so on and so on and so on. Okay, and we're going to get into those in a second. All right. So, sorry, just trying to move on. All right. So, proteins as enzymes. What does that mean? What do they do? They do act as biological catalysts, okay, or enzymes. And there are thousands and thousands of different enzymes in your body. And they play the main role of speeding up chemical reactions. Okay. So, let's look a little bit at some different types of amino acids. Remember, I said there were 20. Okay. And as we can see, these are all non-polar amino acids. But let's look at it. Here's our amine group. Here's our carboxyl group. It's missing the hydrogen right there. And this amine group has an additional hydrogen on there. Okay, you can see this one has a positive charge. This one has a negative charge. So they balance each other out. So overall, it's neutral. Okay, but nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon backbone. All right, we see that with all of these. The only thing that is different, once again, is this variable R group, which is in these boxes. And as you can see, they can be as simple as a hydrogen, and they can get very complex, like this double ring structure we see in tryptophan, which is the precursor to melatonin, which makes us tired. Okay, it's melatonin plays a role in our sleep cycle. But all of these are nonpolar. Why are they nonpolar? Well, if you remember, I said the carbon-hydrogen bonds are nonpolar bonds. Carbon-hydrogen, nonpolar, carbon-hydrogen, 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 carbon-hydrogen. These are all carbon-hydrogen because remember, there's going to be a carbon there and there's going to be a hydrogen coming off of these. And all of these, because remember, wherever there's one of these vertices, there's a carbon molecule and we have to have 4 bonds total. So we're going to see there's going to be 1 hydrogen coming off of that, and so on and so on. So these are all carbon-hydrogen bonds off of that, and that makes it non-polar. Okay. The The other group are the hydrophilic amino acids. And once again, what we can see, we see the same thing: nitrogen, carbon, carbon, nitrogen, carbon, carbon. We see the variable R groups right here. Okay, and if you notice about these variable R groups, is that these have oxygen-hydrogen bonds, and you know that oxygen is highly electronegative, so there's an unequal sharing of electrons. Sulfur is also pretty electronegative, so it has an unequal sharing of electrons. So these are all polar. And again, there's my polar molecule. The NH2 and the oxygen right here also are polar. Same here. So these are all polar, which means that they will be found they can be found in a watery environment. And then we have some that are electrically charged. We have some that act as acids and some that act as bases. Anything that can donate a proton is a acid. This was an OH group. It gave off a proton, so on and so on. But don't worry about that too much. But what's going to happen is an acidic amino acid can bind through an ionic bond to a basic amino acid. All right. So once again, the main amino acid structure is the same for all 20 amino acids. The only thing that is different is this variable R group. Okay, so all of these guys are going to be attracted to water. These guys are going to form bonds with each other. And if we go back to the other ones, excuse me. If we go back to these right here, okay, on the nonpolar ones, these are all nonpolar bonds and whatnot. So these are going to be hydrophobic. Okay, so when a protein folds, we would find most of these nonpolar amino acids on the inside of a protein away from the watery environment, unless that protein is inside a hydrophobic region, like inside the cell membrane. And we'll get there a little bit. Again, so these are all hydrophilic. Why? And the answer was because they have charges, positive, negative, or both. There can be polar molecules. Okay. Remember, the way we build proteins is the same way we build all the biological macromolecules. So what type of chemical reaction is occurring to make this polymer. Just look at it and tell me. All right. Now, proteins are complex structures. They can be just a few. So what is this? Let's just look at this picture right quickly. Each one of these little dots represents an amino acid. Okay, I told you there are 20 different amino acids. This is the amino N, the H2N, and this is the carboxyl N. This is that COOH. So there'd be a carbon with a double bonded to an oxygen, and then another single bond to an OH group right there. Okay, so that would be the sequence of amino acids. And this one is 30 amino acids long. This is insulin, which plays a role in sugar regulation. All right, so this is what we call the primary sequence of amino acids. What does that mean? It means that the DNA, which a gene, the insulin gene, okay, in your DNA holds the instructions for putting these amino acids together in this very particular sequence, okay? And the sequence of amino acids is important because the amino acids are going to interact with each other. The variable R group is going to interact with each other. The variable R group is going to interact with the environment, and it's going to cause that protein to fold in this particular shape. Okay, we'll talk about this in a second. This is an alpha helix. Here's another alpha helix, and these are just straight runs of proteins. It's going to form in a very specific shape. Okay, what we see here with these two cysteines right here, and this one, this and right here, cysteine is one of the only amino acids that has sulfur in it. And when you have two cysteines next to each other, those two sulfurs in their variable R group, and let's look at that. Here's cysteine right here, and there's that sulfur. Those two sulfurs are going to interact with each other and form a covalent bond called a disulfide bridge. Okay, so this is the primary sequence. And what is the primary sequence? It is a sequence of amino acids that's dictated by the gene for that particular protein. So your DNA holds the instructions for building the protein. Okay, and if there's a mutation that somehow disrupts that primary sequence, sometimes the result can be a disease or a disorder that can have some pretty severe consequences. Okay, this is the amino acid sequence for hemoglobin. Hemoglobin has about 130 amino acids, if I remember correctly, and the sixth amino acid in this sequence, okay, there's a mutation. Normally it's glutamate, but the mutation for sickle cell will change it to valine. Okay, this is a polar amino acid, glutamate. This is a non-polar amino acid. So we've changed the properties of the amino acids, and that's going to cause a change in the shape of the hemoglobin protein, which will lead to the sickling of red blood cells. Basically what happens is all the hemoglobin molecules are going to stick to each other like this. Normally they're round molecules kind of floating around within the red blood cell, but when you change that one amino acid, they get a little sticky spot on them. And that causes those amino acids to form a long chain, causing the red blood cell to have this sickle shape, which then does not allow those red blood cells to move through your capillaries very well. Okay. Secondary structure. There are 4 levels of protein structure. First was primary structures, primary sequence, which was the sequence of amino acids. The secondary structure is a result of hydrogen bonds between oxygens and hydrogens of adjacent amino acids in the R groups. And there are 2 basic shapes. There's one called the alpha helix and one called the beta pleated sheet. Okay, and you should remember at this point something about the strength of hydrogen bonds compared to other bonds. Okay? All right, so that's secondary structure. Tertiary structure is the third level of protein structure, and it results in a normally folded protein. okay? And it's a result of multiple different types of bonds and interactions. Here's the hydrogen bonds from the secondary structure. Okay, what this represents, let's back up a little bit here. This purple strand represents the nitrogen, carbon, carbon backbone of many, many different amino acids. Okay, so it's the nitrogen, carbon, carbon backbone of many amino acids, and these are the variable R groups. Here's one coming off. Here is another one. This is a polar amino acid. This is a non-polar amino acid. Here are two cysteines, and there's my disulfide bridge forming. And then we have a basic amino acid and an acidic amino acid. And the acidic ones have a negative charge, the basics have a positive charge, and they form an ionic bond right there. So the tertiary structure is a result of ionic bonds, disulfide bridges, hydrophobic interactions, and hydrogen bonds. And again, this represents the nitrogen, carbon, carbon backbone of all of these amino acids here. Okay. There's that disulfide bridge. That's the strongest of those bonds right there. And again, hydrophobic interaction is a result of the hydrophobic amino acids wanting to get away from water. The fourth and final level of protein structure is not found in all proteins. Sometimes proteins will only go to the tertiary structure or third level, and it's going to be a functioning protein. Other proteins are much more complex, and they're the result of multiple polypeptide. What's a polypeptide? It's just multiple amino acids coming together. It's a primary sequence, okay? So it's multiple proteins coming together to form a more complex protein. So here we have three proteins coming together to form collagen. Hemoglobin, which carries oxygen in your blood, is a result of four different proteins coming together. So again, not all proteins have 4 levels. All will have 3 levels. Some will have 4 levels. The more complex ones will have 4 levels of protein structure. All right, so what can happen to a protein? Proteins work, or especially enzymes, they work in a very narrow range of environmental conditions. They work in a narrow range of temperatures. They work in a narrow ranges of pHs. They work in a narrow range of salt concentrations, so on and so on. And if you change those too much, that protein is going to unfold. And when it does that, we say the protein has been denatured. Okay. Sometimes, if the conditions aren't too bad, that protein can refold. It can renature back into its normal shape. But if the conditions get too hot or too acidic, then that protein will change shape permanently, and it is destroyed. And because it's changed its shape, it will have changed its function. So the question is, what bonds do you think broke first that allow this to happen? Think about those 4 primary levels of structure. We have quaternary, multiple tertiary structures coming together. Tertiary structures, which is a result of multiple different types of bond types, hydrogen bonds, disulfide bridges, hydrophobic interactions, and ionic bonds. Secondary structure, which is a result of hydrogen bonds, and then primary structure, which is a result is a sequence of amino acids, and each one of those amino acids are linked together by a covalent bond. Okay, so which bonds do you think are going to break first? Are going to break first in order to denature the protein? Okay. Anyways, as I said, those bonds can change, and that's how we cook an egg. So an egg has the egg white right here is all made up of proteins. Those proteins are called albumin proteins. They're nice globular shaped proteins. And this is all the yolk itself is all fats and lipids. So when you cook an egg, those proteins are going to unravel. They're going to denature and form long strands. And when they form long strands, they hook up to each other, and they turn this once clearish liquid that the albumin proteins were floating in, turn it into an opaque white egg white. and those proteins are shot. They are ruined. Okay. Um, so that happens when you cook an egg. It can also happen when you, if you ever you ever seen, had your milk go bad sometimes, you might see there's globs of stuff on top of this white liquid. And that's when the milk proteins do separate into curds. You might have heard of cheese curds before. And whey is the liquid that's in there. It's the leftover stuff. Okay, it's kind of nasty, but you can make cheese out of it. All right. So that gets us back down to nucleic acids. And I'm not going to spend much time talking about nucleic acids because we could do a deep dive on those a little bit later in the year. But nucleic acids play the role of storing hereditary information, and they contain the information for making all of the body's proteins. That's what a gene is, okay? A gene is the instructions for making a protein. Well, there's other types of genes too, but that's the main big thing. Two types of nucleic acids. We have DNA, which I like to call the $1 million blueprint, and RNA, the cheap 10-cent copy. RNA is constantly being made and broken down. DNA is protected inside the nucleus, and there's all sorts of mechanisms that prevent it from being changed and all sorts of stuff. So that's your $1 million blueprint, and RNA is a cheap 10-cent copy. Okay, but all nucleic acids are made up of monomers called nucleotides, and all nucleotides consist of three parts. We have a nitrogenous base, which can be A, T, C, or G if it's DNA, or it can be A, U, C, or G if it's RNA. Okay, we have a 5-carbon sugar. In DNA, it's deoxyribose. In RNA, it's called ribose. Okay, there's 2 oxygens down here in RNA and sugar. Deoxy, D means to remove an oxygen. So DNA only has 1 oxygen. Ribonucleic acid would have 2 oxygens, 1 here and then 1 here. And it has a phosphate group. Okay, sorry about the format right there. So here's another cartoon of that. There's my nitrogenous base, there's my sugar, and there's my phosphate group. So nucleotides are also made up of 3 parts. Know what those 3 parts are. Okay, and again, nucleic acids are polymers of nucleotides. There's my 4 DNA bases. Thymine, cytosine, adenine, and guanine. Thymine always pairs with adenine. Cytosine, T pairs with A, C pairs with G. Okay. Do you notice that the pairings are always a single ring structure, which is known as a pyrimidine, to a double ring structure, which is known as a purine. So pyrimidines always pair with purines. In DNA, T pairs with A and C pairs with G. Okay. And there's my bases right there. Nucleotide monomers form chains. Okay, there's my, this is my sugar, my sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate backbone. And I'll show you that in class. And these are my nitrogenous bases. This is one nucleotide, a nitrogenous base, a sugar molecule, and a phosphate. Okay. And again, those are my bases. DNA, of course, is the double helix. It is 2 strands of DNA joined together to form a double helix. I do want you to know these are hydrogen bonds holding those strands together. Purines always pair with pyrimidines, A to T, G to C. RNA bases, it's going to be adenine, guanine, cytosine, and uracil. Okay, and so uracil replaces thymine. And so if you ever see a question, RNA only has uracil, DNA only has thymine. Okay, so this is not the $1 million blueprint. This is the cheap 10-cent copy. There's just have to fix that mistake in that slide right there. Okay, and those are the following bases. In RNA, U pairs with A and C pairs with G. And that's it, guys. That's a lot, but there it is.